We've been looking at what sort of evidence is there for evolution. And keep in mind, at this point, the goal is not proving does evolution happen or not. At this point, the goal is examining where can I see that evolution has occurred? Where can I see that evolution is occurring today? So accepting that evolution is a process that includes natural selection, that we have tools to measure it through. We started by talking about one tool we might use to identify that evolution has occurred in the past is the use of the fossil record, and that we're keeping in mind that we can interpret the fossil record a couple different ways, and that might result in different time frames, so gradualism versus punctuated equilibrium. Then we talked about anatomical structures and the sort of evidence that they provide that evolution has occurred. Very important to keep in mind that homology gives evidence for a common ancestor. So the big thing that we're trying to know about homology is that it does support a common ancestor because the structure is the same, meaning the code to build that structure was the same or similar. That means that the code was inherited from a common ancestor. When we look at analogous or analogy relationships, we don't see the structure being the same. We see the function being the same, and this supports common environment, meaning some sort of pressure in that environment selected for multiple ways to do the same job. When we look at what leads to analogous structures, we do focus on this idea of convergent evolution, meaning different types of ancestors that experienced similar environmental pressures, and this led to similar patterns. And so we have these three different patterns here: divergent, convergent, and parallel evolution. And we did talk a little bit about mammals, especially marsupial mammals, as evidence that there are parallel evolution. Those of you who read some of the gems also saw parallel evolution. Coevolution is another example of parallel evolution. And so now we need to move into vestigial and imperfect structures, and then we're going to really look at the idea of how can we examine all these structures together. So vestigial, we talked about being a vestige, and I just want to give you another example, and that's the example of the wisdom teeth. So if you can think back to humans hundreds of years ago, before dentists, before toothbrushes and toothpaste and floss, before possibly even utensils, when we were cracking nuts with our teeth and gnawing off of the bone. And this is why we have baby teeth, because baby teeth get damaged, and it's a dramatic advantage to have teeth grow back in later, rather than losing those permanent teeth at the beginning of your life and never getting them back. And so wisdom teeth were kind of the third tier, that you might get more wear and tear on your back teeth, and that as long as you at least had those back teeth, you could chew things up, grind them up. And so wisdom teeth were supposed to come in later. Now keep in mind, the average lifespan for a human used to be much, much shorter, talking about maybe living into your 30s and 40s. And so having wisdom teeth come in about halfway through your life gave you an advantage if you had lost teeth in other ways. At this point in human evolution, wisdom teeth are actually no longer needed, and they can be a disadvantage. So really, it's interesting to think that if we allowed people who had wisdom teeth, and their wisdom teeth got impacted, which means eventually an infection, and then we let that infection kill them, we would actually see natural selection occur in humans towards, right, we'd see this directional selection that would show the shift of the advantage being people who have either fewer wisdom teeth, like myself, I only had 2, or no wisdom teeth at all, and people who had a high number of wisdom teeth actually perishing from the impaction. So if we let that happen, then we would see natural selection occurring. Because natural selection has not occurred, because we do surgery and we heal people who get the infection, wisdom teeth remain a vestigial structure, meaning they're a remnant of something that used to be an advantage, but because they're no longer a disadvantage, they don't disappear. The appendix is very similar. We have to keep in mind that in order for them to disappear, there has to be a disadvantage that has to be selected against. So natural selection can select for something or select against. The third type of structure that we have to pay attention to is the idea of the imperfect structure. And this really resides in the fact that natural selection has no end goal. And so one thing that we can look at is the vertebrate, the eye, the idea of how did an eye evolve. And if you go back and you look at lots of different organisms, you'll see even some single-celled organisms that have a protein that absorb light and that help them move towards the light. They're actually photosynthetic protists that have what we call an eye spot. But so what we can think of is the idea that the eye is just thousands and thousands of years of mutations building on top of each other. And so it would make sense that the better the design is, the more you would survive. But it really does happen, the order in which those random mutations accumulated. So it turns out that if we look here at, this is the vertebrate eye, and this is the mollusk eye. So this is the eye that's seen in octopuses. In the vertebrate eye, the nerve is actually behind, the nerve is in front of the receptor, meaning the light actually has to come in and wiggle through the nerve to get to the receptor, and then the nerve signal is sent to the brain to interpret light. And this is not a good design. It leaves the human with a blind spot, which is why we really need two eyes in order to see very well. Versus if you look at the mollusks will sometimes only have one giant eye. And this is because that their receptor is first, and then the nerve is behind it, leading directly to the brain. Versus in the humans, the nerve is first, and then the receptor is behind it. So imperfect structures really support the idea that natural selection has no end goal. It has no perfection idea. So when we look at the anatomy, we do need to keep in mind that we can be misled by what we see. That even though anatomy is what we have historically used to compare organisms, it is not the most reliable, especially when we look at the fact that organisms genuinely change, grow, and develop over time. So the last little part of anatomy that we're going to look at is something called embryology. Embryology is the study of how multicellular organisms grow and develop. And we actually really do need to split this up into two parts. Growth, we know, really has to do with cell division and the addition of more cells. And so how many more cells do we add? How quickly do we add those cells? But then we have the idea of development, and this is really differentiation. And we know that differentiation is truly due to gene regulation. And so it turns out that the more and more data we collected on, especially animals, the more we started to see both similarities and dramatic differences that arose even very early on. So any sexually reproducing organism is going to start as a fertilized egg, and it's then going to start dividing. And what we'll see a little later, as we'll talk about it more, is that the pattern at which these cells divide already begins to show certain relationships. So for example, we'll see lateral division and radial division. You can really see it in this picture that these cells are really dividing in a radial shape, and these are kind of almost adding on top of each other. And we start to see the idea of even more and more traits. The idea is that as we develop, we're going to see certain patterns. So we can use those patterns to help show relationships. And what we mean by these patterns is the appearance and disappearance. So for example, humans actually at a very early part of their life cycle have what are called pharyngeal slits. And they're very similar to gills. They are slits in what are called the notochords, the inside. It's before the epidermis has fully formed. And these slits are actually going to become part of the nerve cord covering. But it's this idea that we have some parts of our bodies that are in common with each other that we really didn't expect. And that's embryology. It's really studying the patterns of how multicellular organisms grow and develop. They can be very misleading. Just like any other anatomy, we need to try to be very conscious and deliberate about how we examine this data, similar to the fossil record. So it turns out that most evolutionary scientists are actually going to look for the least biased form of evidence, and that is what we call biochemical similarities. Bio meaning life, chemical. And so really what we're looking at is DNA and protein sequences. Now we could also examine RNA as well, especially if we're looking at bacteria and humans, keeping in mind that any eukaryotic organism has introns and exons. So really what we're trying to compare is the sequence of either nucleotides or amino acids. And you should be able to talk about why might we want to compare one over the other. Maybe we want to compare both the nucleotide sequence and the amino acid sequence. Maybe we just want to use the amino acid sequence. Maybe we just want to use the DNA sequence. And so you should be able to talk about what the difference is and what implication mutations especially would have here. When we look at these similarities, one of the reasons that this is less biased, it is either the same or it is different. So when we look at this example, we see similarities, and then sometimes we'll see differences. So right here at 1140, we see a difference. The top organism has a G.
Sierra Bodrick, meet your ride in the front office. Sierra Bodrick, please meet your ride in the front office now.
So what we can do from here is actually begin to create phylogenetic trees, or trees that show evolutionary relationships. And we'll get more into this in the next unit. But phylogenetic trees show evolutionary relationships. And in the past, they were usually created using physical characteristics that we, of course, know can be misleading. In this phylogenetic tree, we're just looking at 8 sample organisms, and so we're not trying to say that the closest relationship is between the human, the monkey, and the pig. We're just saying out of these 8, these have the most similarities or the fewest differences. The only part that makes interpreting biochemical similarities a little difficult is the idea of a conserved domain. Conserved meaning little to no change. And so what this means is that some domains or some genes are going to be so important. This gene is so important, so necessary for survival, that any mutation may not survive. And so if you think about certain proteins that are necessary for survival, like DNA polymerase, that's one that's probably going to be a very conserved domain. It is necessary for survival. So we'll create what are called molecular clocks to calculate different rates of how frequently genes have changed. And all of this is evidence that genes themselves have changed over time.